Hi, I'm Dr. Jacob Elmer, a professor here in the Department of Chemical and Biological Engineering at Villanova University, and I'm here today to talk to you about gene therapy, specifically one type of gene therapy called CAR T-cell therapy. Now, what's done in that type of therapy is you extract the white blood cells, specifically T cells, from a cancer patient, and then you genetically reprogram those cells to be able to recognize and kill a cancer cell. So once you do that, you can take those genetically engineered T cells, put them back into the patient, where they will then eradicate the patient's cancer cells. And that's a therapy that's been shown to be very effective in a lot of different cases. However, in some cases, it's not quite as effective. And one of the problems that we've observed there is that sometimes it's difficult to deliver a gene to T cells. And in other cases, sometimes the gene is delivered, but it doesn't get turned on. Instead, it gets turned off. So even if your gene is present in those cells, you have no therapeutic effect because you don't have the corresponding protein that you want to get from that gene, which is needed for the therapy. So that's where my research focuses, is trying to figure out what happens to a gene after it's delivered to a cell. Specifically, the problem that we're trying to look at is that whenever we deliver a gene, the cell interprets that as a threat. It thinks it's being infected by a virus because the virus does the same thing. It dumps its genetic information into the cell. And what happens after that is the cell turns on all of these different proteins that defend it against the virus. So that's where our research is we try to, first of all, see if we can prevent those proteins from getting turned on. But if we can't do that, then try to inhibit those proteins too. And that means that our overall goal then would be to improve CAR T therapy by inhibiting those cellular defenses.